I'm going to be reviewing the process of glycogenesis. Glycogenesis is where glucose is converted into glycogen, a form of stored energy. So we start off with a glucose molecule, and with the help of the enzyme hexokinase, which is used by most tissues, or glucokinase, which is used by the liver, ATP ends up donating a phosphate group to the glucose molecule, which converts ATP into ADP. Glucose is then converted into glucose 6-phosphate because a phosphate group was added to the 6th carbon. The enzyme phosphoglucomutase removes the phosphate from the 6th carbon and places it onto the 1st carbon, making glucose 6-phosphate into glucose 1-phosphate. The enzyme UDP glucose pyrophosphorylase helps catalyze a reaction where UTP, which stands for uridine triphosphate, a triphosphate nucleotide, meaning it has 3 phosphates, is equal in energy to ATP and ends up donating 1 phosphate group as well as uridine to carbon 1, making the original phosphate into UDP, uridine diphosphate, di meaning 2. Since now there are 2 phosphate groups plus uridine, UTP is then converted into diphosphate, while glucose 1-phosphate ends up becoming UDP glucose. The enzyme glycogen synthase helps to catalyze a reaction where UDP is removed from UDP glucose, while the glucose molecules are joined together at carbon 1 and carbon 4. This is known as the alpha-1,4 glycosidic bonds, which can also include alpha-1,6 glycosidic bonds. The cycle ends up repeating as a polymer of glucose molecules begin to form, in which case we call this glycogen.